Hello, everyone. You probably don't know me. I'm fairly new to potato research. I started researching on potatoes about 3 years ago. The project I want to talk to you about today is to do with physiological aging. And as I go through this project, I want you to keep a couple of questions in the back of your mind. I want you to think about, how old are my seed potatoes? If I knew the age of my seed potatoes, what would I do differently? And do I know the impact of physiological age on my business? So you're probably aware that seed potatoes have two ages. They have a chronological age, which is just the age, the time from which they were harvested. And they have this physiological age, which is a measure of the internal state. So my father, he's about 75, I think, and his chronological age is 75. But he did this test at the gym and he bragged to me about it, that he's actually physiologically only 60, right? So humans have a physiological age, right? And that's a measure of sort of how healthy we are and how well our body's functioning. And our physiological age, it's not fixed in the same way our chronological age is. And we don't measure it in the same way we measure our chronological age either, right? We measure it by the health of our body and how well our body's functioning. We measure it on, and it's quite changeable as well. You can actually decrease your physiological age if you treat your body right, and you can increase it if you treat your body badly. And we measure physiological age in humans by using biomarkers and lab tests. So in the same way that our performance as humans is affected by our physiological age, the performance of your seed potatoes also is affected by their physiological age. So on the very far, have I got a pointer? Oh, there we go. So on the far left here, right, we're going from physiologically young to physiologically old. This is what seed potatoes look like as they go through those different stages, right? So when they're very, very young, they don't produce any sprouts. And basically, if you sowed those potatoes, you would get no crop. And as the seed potato gets older, it will start to sprout, right? But it will be only from the apical bud. And then it gets older, it will start sprouting from other places. Those sprouts start to get branches, starts to look really funky like this and form these mini tubers on the mother tuber. And then at this very last point where it's starting to get sort of senile, and this last page is called incubation for some reason, that's when you start forming little tubers directly on your seed tuber. So you don't really want to be planting all the way up here, and you don't want to be planting all the way down here either. You want to be planting somewhere in the middle here when your seed potato has this kind of physiological age. So how is physiological age sort of affected? Well, there's so many things that can affect the physiological age of your seed crop, and I'll just go through these fairly quickly because you're probably aware of most of them, I reckon. But during the crop growth, so that when you're growing the seed crop, the temperature, the rainfall, the light, and the time, so that the cropping season can all affect the physiological age of the seed potatoes. When environmental factors during storage can also affect it. So once you've harvested your seed potatoes from your seed crop, the storage temperature has a huge impact on physiological aging, how long you store them for, the light conditions, the humidity, the oxygen availability. But also there are internal factors that affect the pH of your seed crop, of your seed potatoes. And that can be what variety you're working with and the maturity at which you put it into storage in the first place. There are things that we can control as growers, right? There's the planting date, there's how much we irrigate, what protection we apply, form killing date, and how you handle the seed after harvesting. So all of those things go into making up the age of your seed potato that you eventually want to plant or put into storage. And this is really important because everyone cares about yield, right? Am I right in thinking about that? Yep. So if you plant an old seed potato, right, you'll actually do better if you have a short season than if you had planted a young seed potato. Right, so the solid line here is representing tubers and the dotted line is representing foliage. So if you plant an old seed potato, your yield will go up in this sort of curve. If you plant a young one, it'll follow this sort of curve. And at this point, when you have a sort of mid-length cropping season, you actually get basically the same performance from young and old. But if you have a longer season, you're actually better off planting a young tuber because you'll get a much higher yield than you will if you planted an old one. So, how do you know the age of your seed potato? How old is your seed potato? Currently, there's 3 sort of broad categories of tests you can do to know how old your seed potato is. So the first is these sprouting assays, which is basically where you put the seed potato in an environment where it will sprout, so in the dark, at 18 degrees, lots of humidity, and then you sort of figure out the apical dominance intensity. So how apically dominant is it? Is it mostly that sprout and tiny little sprouts, or is it, has that changed? You can also measure total sprout dry weight to figure out the age. But the big problem is it takes too long, right? There's no good if you receive a seed lot and you want to know how physiologically old it is, and then you do this sprouting assay. It takes you months to do the assay, and it tells you, oh, I should have planted a month ago. It's not very helpful making decisions. It's also not that reliable. Other people have come up with this heat accumulation calculation, and it's calculated using this formula, which is not that complicated. So physiological age equals the time multiplied by the temperature you stored it at minus 4, right? So you sort of accumulate heat units. But this is not reliable, and it ignores aging effects that occur even at 4 degrees, because your tube is still aging even at 4 degrees and below. And it also doesn't consider the timing. So if a tuber is exposed to heat early in its cycle, then it has a different effect to if it was exposed late in its cycle. The third way is using this physiological age index, which is a little bit complicated, and because I've only got a short amount of time, I won't explain it, but it has the same problems as the other types of sprouting assays. It just takes too long to do this. All right, so hopefully I've convinced you that pH is really important for your crop performance. pH is affected by lots of different things that we can't really control that well. Some we can, most we can't. And there's no really good way of measuring it at the moment. So how can we do better? Well, I think we can do better with biomarkers. So just like we do with humans, right? We can measure, in humans, we measure physiological age by DNA methylation tests, for example, by telomere length tests, we do blood tests, right? We can figure out people's physiological age. We can do the same with potatoes. You should know that already seed potatoes have 2 ages. pH progresses differently depending on a lot of different internal and external factors. pH affects yield and canopy structure. I didn't talk about this today, but also size distribution. So depending what market you're interested in selling into, you want to plant seed potatoes of a particular age, otherwise you get them too big or too small. Current pH assays are really unreliable because they take over a month usually. And we're well on the way to developing a new pH assay that will take less than a day, ideally. So there were those 3 questions I asked at the start. How old is your seed potato? And what impact is it having on your business? And how would you change what you do if you did know how old your seed potato was? I reckon you could probably answer those questions in your head. And so based on that, can I just ask all of you a question that I want you to actually put your hand up for? It's a yes/no question, okay? So are you interested, is this the type of research that you would like to see more investment and more research done into? Because we've only just started and I think it's really promising. Can you just stick your hand up if that's interesting to you? Up to. Cool, so like maybe half of you. That's awesome. That's really good to know. Yeah, so that's kind of all I have. What we do plan to do next is to expand this work into other varieties. So we've collected samples from other varieties. This is all on Russet Burbank. We've collected samples from Ranger Russet and also Barossa, but we'd love to expand it to other varieties. And this is all on G0, right? So they're all grown hydroponically. We have got tissue sampled from G3 seed as well. So that's, obviously more commercially relevant because that's what you're going to sow. So there's lots of expansion to do in that realm, but there's also expansion to do in terms of establishing, well, we've got these biomarkers. At what level do they actually make a tangible difference to crop performance? So we establish thresholds. And then my sort of dream is, my vision is that it's part of the seed certification. So when you get a seed lot, it's certified to be, young or old or somewhere in between. That's what I would like to see. Thanks for paying attention.